I cloned an actual human using live sperm DNA. This is the story. Today I wanted to try something new. I wanted to clone someone using freshly donated spermatozoa collected directly from the source. So, I called up a friend of mine who works in the entertainment industry. I told him the plan. All I needed was for him to bring 1 or 2 girls to help. He agreed. I flew 3 hours to Wisconsin, USA. When I landed, he was already outside waiting, standing confidently with 2 women, like this was just another Thursday. I handed him the DNA swab and pointed at the door. Then it began. Lulu in progress. I waited outside, quietly, respectfully, like any responsible geneticist would. After about 24 minutes, he came out. Mission complete. Lulu successfully executed. He handed me the DNA swab. It was fresh, still a little bit warm. I placed it in a chemical grade tube, sealed it inside my protective case, and flew all the way back to my DNA lab with this sample in my hand. That's when the cloning process started. First, I inserted the swab into the DNA cloning kit. I filtered and stabilized the spermatozoa, prepping for extraction. Then, I loaded the sample in the centrifuge to spin out any waste and isolate the pure genetic code. Under the microscope, I analyzed the material. Strong, motile, fully intact, the Lulu worked. I transferred the sample into a sterile test tube rack, then placed it inside the DNA thermal chamber. Heat cycling began. Within minutes, millions of copies of his DNA were replicating in a perfect sync. Finally, I extracted a clear donor cell, removed this nucleus, and replaced it with a duplicated spermatozoa cell. The cell accepted it instantly. Now, this is you at 12 weeks old, and soon I will have you for myself.